Hello, everyone. Thank you for joining us for our webinar today. I am Jyothi Paniyadi, a scientist in the technical support group at ACD. Before we begin, I would like to let everyone know that the audience will be automatically muted for the duration of the webinar. If you have a question, please use the Q&A function to the right of the WebEx window. Please address your question to all panelists, and my colleagues will respond to your queries in real time. In the event we are unable to get to your question, we will follow up with you offline after the webinar. This webinar is being recorded and will be on our website soon. For a quick overview of the topics I will be covering today, I will begin with a bird's eye view of RNAscope technology, followed by general assay workflow and troubleshooting guidelines. I will also talk about how to interpret the assay results, and lastly, list some frequently asked questions. So let's begin by asking the burning question: what is RNAscope? RNAscope assay is an in situ hybridization technique that utilizes a unique double Z target probe design, followed by signal amplification system that allows you to visualize a single mRNA transcript as a punctate dot. RNAscope allows for both chromogenic and fluorescent labeling and is compatible with a wide variety of samples, including FFPE, fixed frozen, and fresh frozen tissues. The growth and adoption of the RNAscope technology is best exemplified by the number of peer-reviewed publications. Since our first publication in 2011, there has been an increasing awareness of our technology, and currently we have over 2,500 peer-reviewed papers published in numerous journals, including many top-tier ones. Also, the RNAscope technology is highly relevant across multiple fields of research, with over 40% of the publications in cancer research, followed by neuroscience and infectious diseases. More recently, the incorporation of the RNAscope technology into single-cell RNA sequencing workflows is demonstrated through the rapid growth of publications using the assay to confirm and spatially map the single-cell RNA-seq data in the tissue context, with over 80% of these single-cell papers in very high-impact journals, including Nature, Cell, and Science. Here is a brief overview of the RNAscope workflow. RNAscope assay is a slide-based assay and is intended for use on sample types similar to that used with immunohistochemistry or immunofluorescence. The assay starts with permeabilizing the sample with the reagents provided in the pre-treatment kit. The pre-treatment enables the unmasking of the target mRNAs, thus allowing the probe to penetrate into the cell and hybridize with the target RNA sequence. The probe is a pool of oligos that are designed using our established bioinformatics algorithm, which allows for target specificity. A series of amplification cascade then amplifies the signal, and RNA molecules are then visualized under the microscope as punctate dots. The signal can also be quantified semi-quantitatively or quantitatively using image analysis software. The primary feature of our RNAscope technology is the oligo-based double Z probe design. Each Z probe has a lower part that is 25 bases in length that is complementary to the target RNA, and an upper region is a 14-base tail sequence that has the pre-amplifier binding site. The upper and the lower parts of the Z probe are linked together by a spacer sequence. Two such Z probes must come together to create a 50-base specific binding site that hybridizes with the target region of interest. The informatics algorithm that is used to design these probes ensures that the oligonucleotide sequences are specific and do not cross-hybridize with any other sequence. The top of the Z is the base for the amplification structure. When two Zs hybridize, it creates a binding site upon which a preamplifier can bind and the amplification tree can be built. A standard RNAscope probe for a target sequence of 1,000 bases or more consists of about 20 ZZ pairs pulled together that are designed to hybridize next to each other along a target region. This allows for a tremendous amount of amplification and signal potential. However, only a few ZZ pairs are needed to bind to the target RNA sequence to generate enough signal for molecular detection. Here is a schematic showing the hybridization and subsequent amplification of the RNAscope assay. The double ZZ probe will bind to the target mRNA transcript. Once the double Z probe binds, it serves as a foundation and binding site for the preamplifier molecule. Once the preamplifier is bound, it serves as a binding site for the amplifier molecules, and each amplifier can further bind multiple labeled probes, sequentially hybridizing to assemble a branching complex at each ZZ binding site. Labeled probes contain an enzyme such as HRP that generates a visible signal after chromogenic reaction, such as with the DAB, that is detectable under a standard bright field microscope. The labeled probes can also contain fluorophores that allow for visualization of the signal under a fluorescent microscope. This signal amplification strategy provides for visualization of target RNAs as a single dot, where each dot represents an individual RNA molecule. If a single Z binds to the target mRNA transcript, the preamplifier cannot bind as it needs both ZZs to provide a binding site. This helps to ensure the specificity of the probes and helps reduce potential background staining. In the double ZZ probe design, we have a tail sequence that is indicative of a channel designator. For example, a C1 probe has a specific C1 tail sequence that will allow only for binding of the subsequent C1 amplifier and labeled probe molecules. Similarly, a C2 probe will have a separate and unique tail sequence, and so on. This slide summarizes all the assays that are currently available from ACD. We have Chromogenic single-plex brown or red assay and Duplex assay on both the manual and automated platforms. The multiplex fluorescent assay allows detection of up to 3 targets for V1 assay or up to 4 targets for V2 assay, and with the high-plex kit, up to 8 or 12 targets simultaneously in a single tissue section. The major difference between the V1 and V2 fluorescent assay is that the V2 assay uses opal dyes from Akoya Biosciences for signal visualization, whereas in the V1 assay, as well as the high-plex assay, the fluorophores are conjugated and are included in the assay kit. We also have some assays available on automation platforms like the Leica-born RX or Ventana Discovery. For RNAscope, both the chromogenic single-plex and duplex assays are available on Leica and Ventana. The V2 fluorescent assay is currently available only on Leica. If your target of interest is shorter than 300 base pairs, it is below RNAscope detection limit, and in such situations, a base scope would be the right choice for you. Currently, base scope is available on both manual and automation platforms. If you're interested in the automation platform, please contact your local account manager. We have our local field application specialists who are available for training you and also troubleshooting on the automation platform. In the next few slides, we will talk about the RNAscope amplification and detection systems for the manual assays. The diagrams here depict the single-plex amplification system. Pictured here is the amplification tree, which is then tethered by an enzyme. On the left is the red assay that utilizes alkaline phosphatase and is visualized by a fast red chromogen. While on the right is the brown assay that utilizes HRP and is visualized by DAP chromogen. A brief overview of the duplex amplification is presented here. The two probe channels used here have independent amplification trees. The channel 2 associated amplification tree utilizes alkaline phosphatase enzyme and fast red substrate for the visualization of the target RNA, while channel 1 associated amplification tree utilizes green chromogen for the visualization of the second target. Please note that our C1 probes by default are ready to use and 1x in concentration, while the C2 probes are provided as 50x stocks that are required to be diluted in a 1:50 ratio. The multiplex fluorescent V1 assay utilizes the same amplification system and is similar to the duplex chromogenic assay. Here, 3 independent amplification trees are built such that the C1, C2, and C3 probes will have their own amplification trees stemming from where the probe has hybridized to the mRNA transcript. The signal can then be visualized from the 3 independent fluorescent label probes. The fluorescent multiplex V2 assay can be used to detect up to 4 mRNA targets. Here, 4 independent amplification trees are built. The C1, C2, C3, and C4 probes will have their individual amplification trees built from the mRNA target. The signal can then be visualized using the opal dyes or PSA plus fluorophores. RNAscope Hiplex allows simultaneous detection of up to 12 targets on the same fresh or fixed frozen tissue sections. This assay is based on ACD's proprietary signal amplification and background suppression technology. Each probe is designed or assigned a specific tail sequence or type referred to as T1 to T12. The target probes, which can be up to either 8 or 12, are hybridized and amplified together. The signal detection is iteratively achieved in 3 rounds with groups of 4 targets. After the first round of detection, the fluorophores are cleaved and the second round of targets are detected. The cleaving procedure is quick and is chemical-based to protect sample morphology and RNA quality. I will now go on ahead to discuss the RNAscope assay workflow. RNAscope assay can either be run in a single day or you may split it into 2 days. If you decide to perform the assay over 2 days, then one option is to complete the sample preparation and pre-treatment on day 1 and then start with the probe hybridization step on day 2. Alternatively, you may finish the probe hybridization step on day 1. That includes 2 hours incubation with probe and subsequent washing with wash buffer, and then leave the slides in 5x SSC solution overnight. On day 2, you will perform the amplification steps followed by detection. The second option is preferable, particularly when you are running the duplex or multiplex assays, as these have additional amplification steps and can take longer to finish. This is an overview of the workflow for single-plex chromogenic assay using FFPE samples. The single-plex assay can take about 8 hours. Please refer to part 1 of the user manual for sample preparation and pre-treatment recommendations. The assay workflow has 4 major steps: pre-treatment, hybridization, amplification, and finally, staining and detection. The workflow for FFPE and fixed frozen samples will include a heat-mediated target retrieval step followed by protease digestion for pretreatment. Fluorescent multiplex V1 assay, also includes 4 major steps, but it is a shorter assay. The 3 amplification steps are required to be performed irrespective of the number of targets you are detecting. In the AMP4 alt step that comes after the 3 amplification steps, you are required to choose any one of the 3 combinations provided, which are AMP4 alt A, alt B, or alt C. I will provide further details on these V1 fluorophore combinations later on during this presentation. Now coming to the multiplex fluorescent V2 assay, as in the other assay types, there are 4 major steps: sample pretreatment, hybridization, amplification, and detection. However, in the V2 assay, during the detection process, the channels are developed sequentially after the amplification step. Unlike in the V1 assay, where the fluorophores are conjugated to AMP4, in the V2 assay, the TSA-based fluorophores are added separately during the detection steps. This offers the flexibility to the user to opt to visualize any of their targets using the opal dye of their choice. One important thing to note here is that V2 assay is TSA-based and requires the purchase of opal fluorophores from Akoya Biosciences. High-plex workflow is performed in 3 rounds, with 4 targets being detected in each round. Round 1 is the longest, taking about 7 to 8 hours, and includes probe hybridization and signal amplification steps. In this round, you will also detect the first set of 4 targets and follow it up with imaging. After round 1 imaging, the cover slips are removed from the slides. The fluorophores that are conjugated to the oligos are clipped off chemically, and the next set of 4 targets are iteratively detected using the same 4 fluorophore types, again followed by imaging. Once all 3 sets of images are generated, they are then aligned appropriately using the high-plex image registration software. Here is an example of recommended sample preparation. With FFPE samples, we recommend fixing with 10% NBS for 16 to 32 hours at room temperature. The recommended sample thickness is 4 to 6 microns, and we also recommend that the tissue is mounted onto superfrost plus slides. In order to preserve RNA quality, it is recommended to store sections with desiccants at room temperature for up to 3 months. For long-term storage, it is best to store at 4 degrees centigrade with desiccant. For fresh frozen samples, please review the user manual for detailed recommendations. A few highlights are presented here. We recommend storing frozen tissue blocks or sections in airtight conditions at -80 degrees centigrade for up to 3 months. The recommended sample thickness is 10 to 20 microns, again mounted on superfrost plus slides. Our assays are also compatible with fixed frozen and cultured cell sample types as well. Shown on this slide are sample pretreatment recommendations for various sample types. To perform RNAscope red assay or RNAscope fluorescent V2 assay, the recommendations shown on this slide are for manual assay platform, and all the sample types require hydrogen peroxide treatment. With each sample type, we are varying the target retrieval time and the protease treatment. For FFPE, we recommend 15 minutes target retrieval and use of protease plus for digestion. For fixed frozen, 5 minutes of target retrieval is recommended, and a stronger protease, protease 3, is used. For fresh frozen, on the other hand, target retrieval is not required, and hence the strongest protease of all, protease 4, is used. Lastly, for cultured cells, again, no target retrieval is required, and the dilution of protease 3 is recommended. Let's now discuss some best practices while running the RNAscope assay. One best practice that we highly recommend is to include controls, both positive and negative, in your assay. Positive controls are used to ensure that the assay workflow is appropriate and also to confirm RNA integrity in your sample. Negative control is used to confirm signal specificity. Single-plex assays use PPIB as the positive control and DAB-B as negative control. PPIB is peptidyl prolyl isomerase B, or cyclophilin B, which is a housekeeping gene, and most novel tissues are expected to show moderate PPIB expression, while DAB-B is from a soil bacterium and should not pick up any signals in samples from other species. For duplex assays, we recommend using a 2-plex positive control, POLARS2A and PPIB, and 2-plex DAB-B negative control. For multiplex assays, we recommend a 3-plex positive control comprising POLARS2A, PPIB, and UBC, and also a negative control comprising 3-plex DAB-B. Similarly, the high-plex assay includes a 12-plex positive control pool and a 12-plex DAB-B negative control pool. Now that we know what controls to use for the various assays, let's see how we can qualify the samples for the assay. If you're working with RNAscope assay for the first time, then we recommend that you run the positive and negative control probes on ACD-provided control slides such as HeLa in order to ensure that the assay that is set up in your lab is optimal. Then proceed to running the assay using the control probes on your experimental tissue to assess the sample RNA quality and to determine the best pretreatment conditions based on the signal detected and the tissue morphology. If your sample passes QC, you can then move on to running your target probe. Otherwise, it is recommended to verify your technique, check for RNA quality, or perform assay optimization. I will be discussing our scoring guidelines in a later slide. Additionally, make sure to have a checklist like the one shown here that can help ensure your success with the RNAscope assay. For example, always work with fresh reagents. Use appropriate assay-specific mounting mediums. We also have some recommended general guidelines such as apply all the amplification reagents in the right order, maintain humidity in the high-DC oven, etc. Here is a typical example of an RNAscope experiment. We used serial sections from a human lung cancer sample and ran a manual chromogenic red assay. What you're seeing is the DAB-B negative control image. As mentioned earlier, this probe is from a soil bacterium and therefore gives the expected clean background in the human lung cancer sample. This is a section from the same sample stained with the positive control probe PPIB. PPIB is showing strong signals that is fairly uniform across the section, indicating that the RNA quality is good. Now, this is a section stained for immune checkpoint marker PD-L1. Here we observe a strong punctate dots indicating expression of PD-L1 in this tumor sample. Based on the control staining, we can be certain that the localized expression pattern seen here is real and specific. Let's now talk about a few factors that can impact your assay results and some tips and tricks to overcome them. In this section, we will also go over the various assay types and the recommended conditions for each of them. This slide shows a detection of PRKCZ in normal mouse colon tissue using the chromogenic brown assay. This is an ideal assay for tissues that have AC-dependent backgrounds such as kidney. But this assay is not ideal when using tissues that are subject to pigmentation such as lung or skin samples. Here is an example of a chromogenic red assay. The image shows the expression of ZAGE1 in stromal cells. The fast red chromogen is used in this assay. We recommend using either eco-mount or vector mount media to best preserve the staining. The fast red chromogen is sensitive to alcohol and if it's exposed, may cause signal diffusion. After chromogen development, the slice should not go through ethanol dehydration, and therefore we recommend drying the slice in a drying oven at 60 degrees centigrade. Here we show a duplex assay for two c-Met positive tumor cores, core 2E2 on the left panel and core 2A2 on the right. c-Met is a tumor antigen, and the signal is seen in red color in both the cores. Core 2E2 on the left is a PD-L1 positive tumor, while PD-L1 is shown in green, while the core 2A2 on the right is a PD-L1 negative tumor, where the green staining is absent. In the chromogenic duplex assay, your C1 probe will be visualized in green and your C2 probe will be visualized in red. A few critical points to note here are sensitivity of both the red and green chromogens to alcohol. Therefore, these should not be dehydrated using alcohol prior to mounting. Additionally, in a duplex, you are detecting two targets and the assay duration is therefore lengthier. We recommend that you consider splitting this assay into 2 days. Coming to fluorescent multiplex V1 assay, this is used for detection of up to 3 targets. Here you can use different AMP4 alternatives, A, B, or C, for the different fluorophore combinations. We recommend using prolonged gold anti-fade mounting media to preserve the fluorescence. RNAscope multiplex V2 assay can be used for detection of up to 4 targets. This is a TSA-based assay, and TSA plus fluorophores or opal dyes are used that are required to be purchased from Akoya Biosciences. Again, we recommend using prolonged gold anti-fade as the mounting medium. Hiplex is also a manual fluorescent multiplex assay and may be used for detection of up to 8 or 12 mRNA targets on the same tissue section. Here too, there is a single probe hybridization step, and this is followed by iterative fluorescent detection in 3 rounds of 4 targets each. Hiplex assay utilizes cleavable fluorophores for Alexa 488, Auto 550, Auto 647, and Alexa Fluor 750. Currently, Hiplex is recommended for fresh frozen and fixed frozen samples only, and as in other fluorescent assays, prolonged gold is the recommended mounting medium. This slide summarizes the general tips that we discussed to ensure success with RNAscope assays. First, follow the protocols. We have various protocols with necessary details to help address the best way to run the assay. Next, sample pre-treatment is very important to ensure that you get optimum RNAscope signal. The pre-treatment conditions will differ based on sample type and may require some optimization. Using controls is highly recommended, especially when you are troubleshooting your assay. And lastly, it is critical to make sure that you have all the components required to run the assay with you before you begin your experiment. The slide shows the standard RNAscope kit configuration. Each kit includes pre-treatment reagents, detection kit, as well as wash buffer. The probes are not shown here. Shown here are some recommended accessories required for running the manual RNAscope assay. The HyBZ hybridization system shown on top right is a critical component of the assay. It is designed to maintain the optimum temperature and humidity conditions to ensure consistent RNAscope staining. Also shown are 2 methods for carrying out the heat-mediated target retrieval step that is required for some sample types such as FFPE and fixed frozen. Our website has step-by-step training videos, which is a great visual guide to assist you with running the RNAscope manual assay. Now let's review some troubleshooting examples and the results after performing optimization on these samples. When running the RNAscope assay, here are a few factors that could affect the outcome of your results. For example, sample fixation may not be optimal, RNA may be degraded in the sample, samples may be drying out, etc. These factors and issues can be addressed by using the recommended sample fixation conditions and instruments. You must also start with the standard pre-treatment conditions outlined in our protocol and then optimize the conditions as required. Here are some potential reasons why you may need to optimize. If the samples are underfixed, this can usually lead to over-digestion of your samples using the standard conditions. If the samples are overfixed, the signal could be weak and you may need to optimize. There are also some special sample types that could require different pretreatment conditions and optimizations. A few are listed here, like liver, muscle, etc. If your tissue of interest is not listed, or you are unsure if your tissue has been fixed as per our recommendations or not, please feel free to reach out to technical support and we can assist you in choosing the right pretreatment conditions. Sample pretreatment is a very critical step for the success of RNAscope. When running the RNAscope assay on your samples, like I mentioned earlier, begin always with the recommended conditions for your sample type. Observe the signal intensity and background with control probes. Also, check the tissue morphology. If recommendations for your sample type are not available in the user manual, it is always best to start with standard conditions. This will help to set a baseline for how the assay performs, and depending on your results, you can then make recommendations to either scale the pretreatment up or down. This slide shows optimum digestion of a tissue fixed per the ACD's protocol recommendation and treated with standard pretreatment conditions. PPIB staining is strong, while the DAP-B negative control is clean. Tissue morphology is intact, and hematoxylin staining is homogeneous. Compared to the previous slide, this image shows a tissue that has been overdigested, in which the nuclei are now undefined due to suboptimal tissue morphology, and the PPIB staining is slightly weaker than expected. Here, the target retrieval time was reduced to 8 minutes, and that improved the morphology and eliminated the background seen in negative control. Compared to the image on the left, you can see that there is better morphology and better signal resolution after using the modified target retrieval time. On the other hand, here are samples that are underdigested. The PPIB staining is much weaker, but the tissue morphology is intact. Here, the target retrieval time was increased to 30 minutes, which improved signal intensity and resolution, and also eliminated trapping of the chromogen as seen in the image on the left. Shown on the right panel is an image of RNAscope performed on a sample that was fixed for too long. As you can see, the signal is very weak. To avoid such issues, we highly recommend following the sample fixation steps provided in our user manual. When a multiplex fluorescent RNAscope assay is successful, you should see images like the ones shown here. This is using the AMP4 Alt-A option and was performed on the HeLa-controlled cell pellet with human 3-plex positive control probes. These are examples of fresh frozen samples where the effect of protease treatment on RNAscope signal quality is demonstrated. In the top two panels are the mouse kidney samples. On the left is the kidney sample that is overdigested due to protease 4 treatment at 40 degrees centigrade. But bringing the protease incubation to room temperature resulted in optimum sample digestion, and you can see clear green and red punctate dots. Similarly, the bottom two panels show examples of mouse brain where optimum protease treatment at room temperature gives a better signal to noise ratio compared to the overdigested sample that shows high amount of tissue autofluorescence background. Tissue autofluorescence is a known issue with some sample types, particularly the FFPE samples. This poses challenges for image analysis. One approach is to use background correction software to reduce autofluorescence. With our multiplex fluorescent V2 assay, since you have the flexibility of choosing the opal fluorophores that you want to work with, we suggest either avoiding the channels that tend to show high autofluorescence or use them to visualize high expressing targets. If you continue to experience autofluorescence issues with your assay, please feel free to contact our technical support team and we are happy to troubleshoot further. To address tissue detachment issues, we recommend using SuperFrost plus slides. If your samples are not adhering to the slides properly, the likely causes may be the ones listed here. We have found that adding an additional baking step can help mitigate this issue. These apply to all assay types. If you're observing sample detachment after hydrogen peroxide or target retrieval treatment, then adding a baking step before H2O2 incubation can help. If you're observing tissue detachment after the protease treatment, then add an additional baking step after the target retrieval. Both baking steps are for 30 minutes at 60 degrees centigrade in a circulating oven. For sample detachment issues with fixed frozen tissues, we recommend drying the slides at -20 degrees centigrade for 2 hours after cryo-sectioning. Add a post-fixation step before hydrogen peroxide and target retrieval treatment, the duration of which can extend anywhere from 15 minutes to 2 hours at room temperature, depending on your sample. In most cases, we have seen that the sample detachment happens after target retrieval. So we recommend adding an additional baking step before the protease treatment. If you continue to see tissue detachment, we then recommend that you omit the target retrieval and extend the protease time to 45 minutes. Also, please contact support and we can help you to figure out the best way to solve this problem. For probe-related troubleshooting, if you are working with 2 scenarios listed here, please contact technical support. For knockout validations, as we have thousands of probes currently in our inventory, we would like to help ensure that the probe you are picking is specific for your wild type versus knockout validation. If you're working with a novel gene and no catalog probe is available, please feel free to contact your area account manager or visit our website to get your custom probe design feasibility test started. We provide recommended sample preparation protocols for most commonly used sample types. If you're working with unique sample types, such as whole mount, free-floating, or bone, please feel free to contact tech support for suggestions and guidance. One of the most asked questions is, can we combine RNAscope ISH with immunohistochemistry or immunofluorescence? The answer to this is yes, you most certainly can. Both ISH and ISC procedures have similar workflows as shown here. Both require sample fixation, some kind of pretreatment to unmask your RNA or protein, and subsequent detection. The recommended workflow is shown here. You would pre-treat and permeabilize your tissue or cells first. Depending on the tissue, this may require both a heat-mediated target retrieval as well as a protease digestion. You would then complete the RNAscope ISH portion first and then follow with the ISC staining after. However, since RNAscope includes a protease treatment step that can diminish the protein signal, we suggest that you empirically optimize the protease treatment in order to achieve a good balance between your RNA signal and the protein signal. Here is an example of using the RNAscope single-plex red assay to detect the PDL1 probe with subsequent IHC staining in blue for CD45 protein. We have so far talked about how the technology works and the various assay workflows. Now let's discuss how to analyze the assay results. RNAscope staining can be quantified by manually counting the number of dots per cell. This is also known as the semi-quantitative scoring method. With this method, a score of 0 to 4 is assigned based on the average number of dots per cell. We have a guideline document on our website that you can download that describes the different methodologies to quantify your results. If you have further questions, please contact technical support for recommendations. Some questions that you may have at this point are, what does a dot mean? What is the significance of a dot size? And what is the difference between a dot and a cluster? In this image, where an RNAscope chromogenic red assay has been performed, the signal appears as numerous punctate red dots of various sizes and intensities, and also a few clusters. It is the number of dots and clusters that are important rather than the size of the dot or the intensity of the dot. Each punctate dot, as shown by the blue arrow, represents one mRNA molecule. The slightly larger, darker red punctate dot shown by the yellow arrow is also one mRNA molecule. However, it is larger in size as more number of ZZ probes have bound to the target molecule. In contrast, a cluster shown by the brown arrow is oblong with a slightly irregular border and is the result of overlapping signals from multiple mRNA copies. Similar patterns of dots and clusters are seen in the fluorescent versions of the assay as well. The yellow arrows point to single mRNA molecules with fewer ZZ probe pairs bound to them, compared to the dot shown by the teal arrow. The white arrow is pointing to a cluster with an irregular shape due to an overlapping signals from multiple mRNA molecules. A quantitative way to analyze RNAscope data that is more objective and rapid is by using automated software analysis platforms such as the Halo software system from Indica Labs. As mentioned earlier during this presentation, we have over 2,500 peer-reviewed publications that used our RNAscope and BaseScope technologies. Many of these papers, either as full text or as abstracts, are available at our website. Please visit the link provided to view and access this publication list. With this, we come to the end of this presentation. We will continue to be online for a few more minutes. Please feel free to send in questions to our panelists who are waiting to answer your queries via the Q&A window.